So, uh, as you have seen in the previous part of this lecture, that in the first part, whatever the k tuple or the small sequence or small words are there, that is compare, and we'll be just narrow diving the one leg sequence to, for example, some 10,000 sequence for the comparison. So that would be your first step. Now after that, you can see that you can just have it matches the identical word for each list and then creates a diagonal by joining the adjacent match, but it will only count the non-overlapping word, and it then score the highest scoring region using the replacement, which is known as the init one, and for that you can utilize the palm 250. So in this you can see that first of all what I am doing is that whatever the 10,000 sequences are matching with this my sequence, for that I'm just giving the score of the palm. So by using the palm matrices, whatever the 10,000 sequences are matching, that would be also ranked. And whatever the first ranking sequences would be there, that is normally known as the init one. So for example, by this again from the 10,000, I'm selecting the 1,000 sequence for the further comparison, and this 1,000 sequence whatever I have taken for the comparison, that would be just going for the further alignment. So whatever the word we have compared, besides that word, whatever the matching portions are there or not, that would be getting matched. And after that, from the 1,000 I can narrow down the sequences to the, for example, some 100 sequences, and for these 100 sequences I can put some kind of the score. So by this mechanism, this FASTA comparison is normally done. Now FASTA is more or less outdated. People are not using the FASTA sequences, but just historically we have to use the FASTA. So this is the second part what we have already discussed. It then tries to join together the highest scoring diagonal and then allowing for the gaps, and the best score from this is known as the init n. So init 1 would be your 10,000 to 1,000, and init n would be your 10,000 to the 1,000 or 100, whatever you are taking. Finally, it makes an optimal local alignment around the region. It is discovered by using the Smith-Waterman algorithm, and this is the last alignment step. It is applied only to the small number of the sequences, which are known as the hit sequences, and this had the high init n value after the database search. So when you are just running the practical of this, you might have gone for the entire process. So that would be getting completed by only 2-3 clicks. But logically, you have to understand that whenever I'm just going for the 2-3 click, what is happening behind the computer. So these are the different kind of the FASTA programs which are developed along with the time. So you can just have the simple FASTA, which is already we have discussed. Then you can have the T FASTA. So that will compare the query of the protein sequence to the DNA sequence database. After that, the DNA sequence database has been translated into the all 6 reading frames. Why such kind of the things are required? Because many times it will happen that suppose you compare the sequence finding process, then to find the sequence of the protein, it is very difficult in comparison to find the sequence of the DNA. So that is the reason that you can see that wherever the DNA sequence are there, that would be large number would be there, but the protein sequence would be always less. But now with the help of the computational power, it is possible to convert the gene sequence into the protein sequence computationally. So even though I'm having the any part of the sequence, protein or DNA, I can just go for the vice versa comparison, right? Similarly, we are also having the FASTA-F, so that will compare a set of ordered peptide fragments obtained from the analysis for a protein by cleavage and sequencing of the protein band resolved by the electrophoresis against the protein database. So this is like your peptide fingerprinting, only the peptide would be getting matched. Same way you can also have the so many other kind of the variations, but people are using this FASTA and T-FASTA most commonly. So these are the few other kind of the alternate, at least you can remember the name of that for your understanding. Now we are coming to the second part that we have seen the FASTA. Now we are just coming to the second kind of such comparison tool, which is known as the BLAST. So the BLAST is basically a basic local alignment search tool. In short, that is known as the BLAST, and that was discovered by the Eldridge in the 1990, and that is the heuristic method for the local alignment. Now what is done inside that is it is designed specifically for the database search and based upon the same assumption of the FASTA that take the good alignment contain a short length of the exact matches, and both FASTA and BLASTA search for the local sequence similarity instead of having the exactly the same kind of the goal. Though have used the software with the different algorithm and the statistical approach. The blast benefit with comparison to the FASTA is FASTA, even though the word FASTA is there, but computationally that is the slow software. In comparison to that, blast is the rapid or the speedy software. The blast is user friendly and that is statistically rigor, and it is having the more sensitive kind of the nature. So that is the few detail about the blast. Why we are using the blast? So finding the model organism for the study of the disease. Suppose we want to study the entire human disease. So we want to compare that which is the best suited model organism for that. So yeast can be used as a model organism to study the cystic fibrosis. So such kind of the question when you want to ask or when you want to solve, you can use the blast kind of the search. Suppose we are taking the same example further, that cystic fibrosis is a genetic disorder that will affect the human. If yeast contains the protein that is related or the homologous to that protein involved in the cystic fibrosis, then yeast can be utilized as a model organism to study this disease. And the study of the protein in the yeast will tell us about the function of the protein in the human. So whatever task is that we want to find out a protein which is present in the human being, and we want to find out the similar homologous protein which is present in any other organism which can be utilized as a model organism. So what we are trying to do is we are not comparing the entire sequence, but we are just comparing the some part of that sequence, and we want to find out the homologous sequence. So to answer such kind of the questions, we can utilize the blast kind of the search. So this blast kind of the search that would be just used for this purpose. So that will find out the homologous genes and the proteins. Homologous means which is having the same kind of the origin. So have a common ancestor that are related, having the similar kind of the structure and having the similar kind of the functions also. How the blast is basically working? So blast is again, as I told earlier, that is the local basic local element search tool, and that is made available by the NCBI Gene Bank. So that is available freely on this tool. Even the offline version of the blast is also available, but for that you have to create your own database. Blast, like FASTA, is a word based method. However, one the major difference is the blast require the pre-formatted search database. Pre-formatted search database means your database should be properly formatted and it should be there in the standard format, right? So that is always required. So that is the requirement of the blast search. What would be done inside that is, so input and output. So in this you can see that input query sequence is the Q and the database sequences are the DB, and we'll be just going for some kind of the minimal score which is known as the S, and output would be the sequence from the database such as Q which is having the sequence score higher than the S. So this is like that for example I'm taking your examination in the class and I'm declaring that those who are having the more than 40% students, they are passing the examination, and those who are having the less than 40% that are not passing, right? So like that we are creating some threshold and we are just selecting the sequences which are having the score above the threshold value. So Blast Search, it is using the GenBank. So this Blast web server let you to compare your query sequence to the various sections. So these are the sequence databases where you can compare when you are using the Blast server. The most common kind of this database is utilized. There is the non-redundant database that is known as the NR database. NR database means in that all the same kind of the sequence are getting removed. So that is known as the non-redundant database. Then we can also have the month, we can have the RefSec RNA, you can have the RefSec genome, EST, taxon, protein, PDB, and lot many sequence you can get in the option, and every day it will get changed also. So depending upon your need, you have to select the database which you want to compare. As I told earlier, NR is the most common kind of the database to compare. Blast step. So Blast is also working in the 3 steps. So it is also using the word-based method like the FASTA. Similarity matching of the word 3 amino acid you can take, or you can take the 11 bases if you are taking only the ATGC sequence of the DNA. Does not require the identical word. If no words are similar, then there is no element and will not find the matches for the very short sequences, and does not handle the gap well. Gap basta is somewhat better. So it will work in the 3 step. It will find out the list of the high scoring word. Then it will compare the word list to the database and identical exact match. And for each word, match the extent and the alignment in the both the direction and find the alignment your score which is having the greater value than your threshold value hash. So it will start like this. So the first step what we are supposed to do is, for the query, find out the list of the high scoring word of the word length L. Suppose this is my query sequence which is having the L, and suppose I want to compare the word, then what I can do is maximum L minus W plus one word. Typically W is the 3 for the protein. So instead of the comparing the entire sequence, what I'm doing is I'm just comparing the word which is coming out from this. So this is the word list, and this word list I'm just comparing. So from each word from the query sequence, find a list of the word that will score at least T when the score using the pay score matrix is PALM 250. So that would be the first step, that is more or less similar to the first step. Now how BLAST is working fast? So you can see that it is working fast like this. So it will break the query into the word. So these are the query which is created into the word. At the same time, instead of comparing this one word with the full length sequence, on the other side it will also create the database also into the word. So it will break the database sequence also into the word. So you can see that instead of comparing this entire sequence with this word, what I'm doing is whatever the database sequence is there, from that also I'm creating the word, and after that whatever we are doing is we are just comparing the word. So you can see that compare the word list to the database and find out ideally exact matches. So I can see that whatever the word list is there, that is matching with this one sequence at only one place, means it is having the matching of only one word. Over here that is not matching at all, and over here you can see that that is matching at the two places. So again you can recall the simple example that from the Rajesh Kanjibai Patel, instead of going for three word, for example in this I am creating only two word, Raj and Pet. So in the first sequence you can see that there is a matching sequence which is having the Raj, but it might have the Rajeshri Patil or something like that else would be there. I can have the other sequence in which Raj is not there and even Pet is not there, so that I'm totally removing. And at the same time I'm having the sequences where I can have the Rajesh Patel and I can also have the Rajesh Patil, I can have the Rajeshri Patil or whatever it may be. So like that you can find out that words are that can compare over here. So exact word, suppose I'm getting the more word, it means that the sequences are more similar to match, right? So after that, it will find out the location of the matching word in the database sequence where it is getting matched, and according to that only the result will be shown, which is we have seen earlier. Then third thing is that for the each word match, extend the alignment in both the direction to find out the alignment that score greater than the threshold value S. So what we are doing is individually we are just enlarging the alignment on both the side, and after that we are just giving the score. And after giving this score only, the computer will find out that above threshold value this many scores are coming. So that score would be directly taken. So instead of comparing each and every part for the further, this scoring would be the base for the comparison. So you can see that this is the places where it is matched. Use the two word matches and anchor to build an alignment between the query and the database sequence. So that will fix the position over here only, and then scoring of the alignment would be done. So that is also known as the extension. So one search space is seeded and the alignment generally generated by extending in both the direction from the seed. The example you can have is the quick brown fox jump over the lazy dog and the quick brown cat goes when she sees him. So you can see that in this some places are getting matched, and after that only it is not getting matched. So can align the first six characters and how far we should continue. So that will just give you the condition like this, that suppose you are comparing this, then you can find out that the quick brown fox up to this it is getting matched, after that that is not getting matched, so you can just drop off the score after that only. So this is a way by which you can just go for the extension and you can have the some kind of the score out of that. Then high scoring pairs are getting aligned region. So whatever the scoring we are doing, after that we are just aligning, just we are shorting them, and we are just selecting the scores which is getting, which is selecting the very score high rating pairs. So which is known as the high scoring segment. In short, that is known as the HSP. And the BLAST then produce the several short HSPs rather than the single aligned region. So that is the way it is going to the compare the other part, right? So this is thing which is done. After that we are having the different kind of the score in this. So the BLAST is having the one basic score which is known as the bit score, and that the second score is there which is known as the E value, right? So this score S that is derived from the row alignment score. So bit score S, it is coming in which we are just getting the statistical property of the score system. You have to take in this by normalizing the row score using this formula. So there is no need to remember this formula and other, but at least you can remember that the bit score or the row score is known as the S. After that, the important part is coming, that is known as the E value. So E value is the score which is depending upon the probability. So why this is important? Because when we are comparing the sequences without any match or without any kind of the homology also, some sequence would be similar. So how to find out that whatever you are getting that is statistically significant or not? So for that we have to take the E value. So the lower the E value and closer it to 0, higher is the significant of the match, and the sequence element which is having the E value of more than less than 0.05, it means that the similarity has 5 in 100 chance of occurring by chance alone, right? So this is a way by which it is including the probability, right? Now this E value, why it is important? That E value is like this, that for example my son is coming to me and he is telling that daddy I score third number in the entire class, so I should be happy or not? So first of all, I should know that what is the class size. Suppose the class size is only 3, then my son is at the last position. But suppose I'm finding that my class size is 100, then I can tell that my son is coming in the top 3%. So like that only, whatever the score is getting normalized with the database size, and even the probability of that is also getting changed. So that kind of the additional parameters can be coming whenever we are using the E value in the analysis. As you have seen the different kind of the alternate of the FLASTA, similar to that even the BLAST program is also coming in a different kind of the flavor. So you can just have the BLAST P, so that is compare the protein sequence with the protein sequence. You can have the BLAST N, that will compare the nucleic acid sequence with the another nucleic acid sequence. That would be BLAST X, that will just convert the nucleic acid sequence into the protein sequence, and then it will match with the protein sequence. So that is known as the translated nucleic acid sequence. So normally whenever we are going for the annotation of any genome, we are using the BLASTX, because when we are running the whole genome sequence analysis, we are getting the sequence of the nucleic acid, but that sequence of the nucleic acid that should be matched for that function with the protein database. So over here we are using the BLASTX. Similar to that, we are also having the T-BLASTN, in which the protein sequence is converted into the nucleic sequence, and that is matched with the translated nucleic sequence. Same way you can also have the T-BLASTX. Among this, the upper three are most common, which is normally utilized. Besides that, even we are having the PSI blast, so that is the position specific iterative blast. So an iterative search is done along with the blast algorithms, and profile is getting matched. So these are the few kind of the alternative of that, and these are the few advanced kind of the blast system you can take. In this one is coming which is known as the pi blast. So in this what is done, your database sequence is there and what query sequence is there, and it will search for the pattern in the database sequence. So it will be just searching for the query sequence, it will find out the prosite pattern, and it will just go for the sequence database. So in this what is done, instead of comparing the entire sequence, the prosite is a same kind of the protein which is getting same kind of the portion of the gene which is getting repeated, so that would be getting matched. So apply score limited to the dynamic program from both the end and for the pattern search. So this is a advanced kind of the blast search which is normally utilized. Same way we are also having the PSI blast. In the case of the PSI blast, what we are doing is whatever the query sequence is there, that is matched with more than one sequences, and that sequences are created as a profile, and this profile would be matching with the other kind of the sequence. So what we are doing is the query sequence, it will be matched with some of the sequences, and that multiple sequence alignment and profile would be done, and that profile would be matching with the other. So this is like that, that for example you want to come to VNSU, somebody want to come in VNSU and he want to find me. So one condition is that he should know me completely, that the Rajesh Patel Department of Bioscience and he's serving as a professor is sitting in this room. If he is having all the information, he can directly come to me and meet. Other alternative is that he is knowing that my name is Rajesh and I'm just serving in the bioscience department. So what is happening? He is now having the two options, that on the campus there will be so many Rajesh that might be serving in the different department, and at the same time in the bioscience also so many persons would be there, but at least in that my presence would be there. So instead of comparing only the Rajesh Patel bioscience, now what option I can do is I can create the profile of all the Rajeshes and all the persons which are serving in the bioscience. And after that, when I'm comparing both these things together, I can find out easily the Rajesh Patel which is serving in the bioscience department. So that is a way by which this position specific related blast is working. This is the side blast, whatever you see, why, where it is utilized. So the basic application of that, it includes the distance homology detection, fold assignment, domain identification, evolutionary analysis, sequence annotation, function alignment, profile export to the other program, sequence clustering, and structural genomics target selection. So instead of going for the simple comparison, suppose I want to go for the advanced comparison, I want to find out some portion which is common between the two protein, which is known as the domain. Or suppose I want to see the specific kind of the fold which is present or not. So for that purpose, this side blast is basically utilized. So these are the summary of the blast related programs. So now even besides the blast also some other programs are there, and that is the RPS blast, hammer, SAM, and PSI grade. So these are the some kind of the softwares which are can be utilized. So this is the sequence alignment programs which are getting developed. So you can see that in the 1970 this Needleman has developed, 1981 Smith Waterman has came. After that we had this FASTA, Blasta, then so many other softwares are coming. So like that in the 2000 we had the even BLAT also. So right now we are not going in the BLAT, but even the blast is not having that much speed. So to increase the speed, people have also developed the BLAT and other software. So these are the things. Now suppose we want to compare the FASTA and BLAST, then you can just have the comparison like this, that the FASTA that will search for all matching of the word of the length k, and BLAST searches for the most unusual high scoring word protein in the length of the entire sequences. FASTA that will calculate the statistical parameter from the unrelated sequence during the database search. BLAST to calculate the parameter for the scoring matrices game penalty combination, and that would be just more speedy with the compare. So low complexity region can give the high scoring matching with the sequence and that are not related to the query sequence, and FASTA 3 does not remove the low complexity region, and BLASTA 2 provide the facility to remove the low complexity region, because we have seen that in the case of the eukaryotes some of the portions are getting always repeated, and this repeated portion would be also creating some kind of the comparison. So suppose I'm running the FASTA, then I can get the score even with this low complexity region or the repeated sequence, but in the case of the BLAST I can remove that kind of the sequences, right? So this is a thing, I think we have to just stop over here only. So this is up to Blasta and Fasta. In next lecture we'll go further for the multiple sequence alignment.